And uh, yeah, actually, that's a good idea. Uh, so today's chat and discussion will primarily be about limitations and measurements in the ocean and why oceanography is important and why cost is critical for measurements. We're going to do this in a fun way because Enrico and I are on a boat right now in the middle of the Pacific. So I'm just going to give a quick tour of the boat, basically. So the first 5 minutes is me walking around showing you kinds of instruments that are there, and then I'll transition to talking a lot more about the challenges. And since time is short, I just want to get started. So I know people haven't joined in, but we're recording this to begin with. And then interrupt me anytime with sets of questions. So first of all, the ocean is big. To prove that to you, you can look out there. And, you know, one of the things that you realize is we are surrounded by an endless amount of water. We will talk about that in context of statistics and other things, but there is no sense of thinking about the scale of this problem, but by immersing yourself in a vessel or on the water. So I'll kind of just walk around and get a sense. Uh, we are on a UNOLS boat right now, which is a NSF boat essentially. NSF has a fleet of around 13 active boats. So this is one of them. The ship is called Sally Ride. You can see it's a massive ship, massive deck. There are two just quick instruments I'm going to show, which are on the deck right now. One of them is this instrument. It's called Mockness. We'll talk about this in a second. But primarily what you can see is a giant frame, which is a meter by a meter, that goes down all the way to 1,000 meters. I mean, it can go even further. We usually go down to 1,000 meters. This is what we use to collect samples. The second critical instrument is a CTD that you can see right there. Neil, the marine tech, is working on it with lots of little bottles, and you're going to see these bottles in a second. These bottles allow us to sample water at different depths, and we use these sets of sampling techniques to figure out what's living far deeper in the water column itself. And I think I'm going to give you a quick tour of the lab, and then I will settle down in one place, and we'll kind of start discussing why, although this infrastructure is very cool, is very limiting because very little can be studied and how expensive it is to do this. So I'm not going to do the entire lab. I'll just show you our lab very briefly. Hey, Hannah. I'm going to show the student what the lab looks like. So it kind of looks like your lab, wherever you might be. And of course, lots of measurement tools here that we will talk about in a second. But one of the nice things about these large vessels is that we can bring many traditional instruments, lots of control panels that allow you to essentially map and measure and control the deployment of some of these instruments. I am going to now walk and sit down at a place that I'm not just, it's a little bit rocky today to go sit someplace. So any questions so far? And I'm almost there. Just one more minute. Okay. Finally, it's quiet and stable now. I'm gonna sit down and connect. Okay. While I connect, any questions in what we just saw?
Maybe to kick us off, what are you most excited for about this trip?
Uh, I think this trip is really focusing a lot on the dark zone in the ocean, which is called what I'm just gonna let me connect through my computer in a second. So, because that way, much easier to see everyone. Okay, so I'm connecting online. I'm gonna go offline on this machine and come and pop back up on the second machine. So just 1 minute. Okay. Um, two very important things for this trip. One is really the mission is called Dark, and it's really about measurements and life in the what is called the twilight zone, or the dark parts of the ocean, which is 500 meters and below. So we know quite a lot about surface oceans, but we know very little about uh, any cycles around any nutrient that you can choose, any sets of elements that you can choose in the deeper parts of the zone. So that's one. And then the other experiment that I'm very excited about is we are testing a novel idea that's been proposed last year of using, again, in tabletop experiments on the boat, clay and natural clay as a carbon sequestration mechanism. So could, just like you might have heard of that Sahara Desert pumps a lot of dust, including iron, into the ocean, uh, how does that dust interact? And last year there was a paper that proposed this idea from a group in Dartmouth, Muckel's lab, that clay can induce faster sequestration of carbon. So we're about to test that idea at a kind of a more from a microbiological point of view. Why does something like that has a potential to uh, we'll find out. So those are sort of two. There are many other things that we're trying to do, but just to give you a context. And of course, any of these sets of things can only be done on a vessel, primarily because you cannot recreate much of the biology that's here to begin with. Okay, any other just anything about what people just saw visually? And then I'm going to transition and do this in a much more structured way so that we can lay out the plan for today. But just I wanted to give you all a tour because a couple of the next classes will be on Zoom, and I'll show little bits and pieces of ship's life. One ship's life is that's my bed. You get to sleep on a bunk bed, which I really love, but I didn't sleep that much last night because the ship was rolling. So the best strategy is to strap yourself to the bed when the ship is rolling. Okay, any
Manu,
questions? Go ahead, Danny.
quick question. You showed some interesting imagery of microscopes and things. Do you have any tanks for when you have the water, if you have something interesting living in it to look at it, but then you put it back in the tank if you want to keep it alive? Or do you just
Yeah,
take
so
all the
we have
water out and look around underneath the microscope?
yeah, we have two what are called incubation rooms. One of them is set up at 10 degrees Celsius, another at 4 degrees Celsius. These are walk-in refrigerators. So we do a lot of work as a lab isolating creatures and trying to keep them alive. A lot of other labs that are working and folks focus much more on processing their samples, filtering them, and then doing analysis post. We as a lab focus on live physiology because most of these animals cannot be kept alive. So we actually do have tanks. We also have a larger tank, which we call, talk about frugal oceanography. It's basically a hot dog roller. I'm assuming you've all seen hot dog grills, but there is a thing called hot dog roller, which also rolls it. But instead of putting hot dogs, we put our incubation chambers, which are just cylindrical vessels in it, and they keep rotating. So you get continuous mixing, and that's how we keep cultures. So any native microbial cultures we keep alive using that as an incubation. What is rare to have is a 4C walk-in room. So that's super nice because nothing will live. The moment you take them out, if you don't maintain them at the same temperature, light, everything dies pretty quickly. The incubation rooms also have red light so that phototoxicity is a huge thread in most life below the 500 meters. So you have to really make sure you don't expose them to wavelengths that they're not used to seeing. Any other question? And then we're going to jump into kind of uh, just a brief discussion on what we discussed last time. And then I want to do another round of scaring you all about what's coming on the oceanography side. Okay, so let me switch gear and let's do this in a little more structured way. And then much of what you just saw will make sense in a little bit. Okay, before we start, I think one kind of a homework that's coming up that I'll describe on Monday. So Monday's lecture will really be about doing back of the envelope calculations for the problems that you're interested in. We've introduced 3 problems so far. We've talked about malaria, we've talked about schistosomiasis, and today we'll focus on just the general problem on measurements in the ocean. After Monday's lecture, we'll have the first kind of a team exercise, which is you will choose a specific problem and you'll try to tease apart and understand its scale. And from that, try understanding what should be the cost framework for solution for that scale of a problem. But I'm just giving you this as an upfront hint. We will talk a lot about numbers today. We talked some numbers around malaria. We talked a little bit about numbers in schistosomiasis. But on Monday, we're going to do a very deep dive on how to estimate and what type of data sets are out there that are publicly available for you to estimate the size of a problem that you're trying to solve. And then there will be kind of a team homework. This would be the first time that you all start working in teams. Also, Monday, the recruitment forms are out now. So around Monday, we'll also announce the international student teams itself as well. So we'll do quick intros with them too. Okay, unless there was any other question on malaria and schisto discussions that we had, I'm gonna jump into oceans. Not literally jump, because that is illegal, by the way. Although it looks really pretty outside, it's super dangerous. And it used to be in the old days from Unal's boat, students could actually take a swim. Somebody got killed by a shark, and since then, rightly so, it's actually banned. You cannot jump in the water. So, any comment on malaria schisto thread? Anything that struck people's mind? Did it bother you enough to kind of think a little bit more about it? Any comments on kind of the, I don't know when it was, no, Monday's discussion?
I think it's frustrating that we're moving away from the problem. Like
Yeah,
lack
I'm
of
I'm I'm
funding.
happy you're expressing that frustration because although I didn't talk too much about it, I think things are getting worse and have already gotten worse. So it's a little bit ironic in many ways with the scale of the problem and many of the yeah, current circumstances make a picture far more bleak than what I had even painted in some sense. Um, so it's, I think in person we'll talk a little bit much more in granularity about it. Would actually be really valuable to just have some of the discussions around that, especially for global health. I think climate work is starting to be impacted, but the global health work has already been massively impacted. And so I think it's important for you all, just as a homework, read as much as you can in that space so you have a better intuition around, uh, you know, the granularity of impact. Because then again, we are all also voices for science. I was reading this article today that science doesn't have a trust problem, it has a voice problem. And what it was implying is that, I mean, we say that, oh, people don't trust science, people don't trust science. Uh, and the article was arguing that it's the fact that we don't have a unified voice. 300,000 scientists are supported by NIH, but less than 1% or so would even say anything because everybody's afraid of saying anything with what's happening with the NIH currently, for example. Today morning is the news that NSF, how many of you are on NSF graduate fellowships? Anybody? At least some of you have applied for it, right? NSF just decided to cut them by half. So anyway, that it just came out as the news. Yeah, I think I'm very happy, Noah, that you're mentioning this. Will kind of, it's valuable to have this conversation in a much more greater detail, actually. But it would actually be also worthwhile doing that in person. Okay, let's just talk about oceans per se. So just as a, for me to know, I remember Danny is in the oceans department. Anybody else who's sort of ocean affiliated in some way or not yet, right? Okay, so I think my hope is that by the end of this lecture, some of you say that you are oceans affiliated. And at least for the problem space, early on, I like doing some of these grand problem discussions because even if you have no background and before taking this class, you weren't thinking about this problem, it would be fantastic. There are two ways of approaching a good project to be thinking about. One is that you have an incredible expertise in one domain, and then you're bringing it to a field that you know very little about. Or sometimes the other way around, that you really deeply understand the problem very well, and then you partner with people that bring a completely new skill in the sense that suddenly new ways of thinking. The best case scenario is when both of them is present, but just as a team, it's valuable. And it's perfectly okay if you identify yourself as one type of a person versus the other. But both of this are critical. Okay, so with that as a background, let's just talk about uh, what's the measurement problem in the oceans per se? Why has it been so hard? Lots of statistics get being talked about. And I'm curious, it's uh, for some of you who might not have seen the open ocean explicitly, um, what kind of, are there analogies that come to your mind when you think about the grandness of the scale of the problem? And what I'm defining as a problem here is, you know, when we say we study the ocean or we need new ways to study the ocean, any analogies that come to mind? What do people think about when they look at this beautiful picture, for example? Uh, any fields that you can make analogies to? What does it feel like when you're kind of in the middle of this vast body of water? Yeah, go ahead, Danny.
I think about like laying out in a field and looking up and looking out into outer space, I think is and the vastness, the scale, the unknown, I think is the closest that I find inspiration from when I think about the ocean.
Yeah, I think space is a phenomenal analogy to it. And frankly, in some sense, people make that analogy very strongly. Everybody says that, oh, we know more about the surface of the moon than the earth. And these types of analogies, although you can rigorously debate them, they are important. And it is true that the resolution that we can map the moon is much higher than we are mapping our bottom of the surface of the ocean. Any other analogies that come to mind? Other similarities, other challenges that you might have to face if you are designing for making measurements in the ocean? If you were building something that has to work in the ocean, what do you think might be problematic or difficult as a challenge?
I can say that the cost of getting to the middle of the ocean is already a big hurdle.
That is a fantastic analogy. So I didn't mention this, but Enrico knows this. Uh, we are spending right now $80,000 a day to be right in the middle of the ocean. So let me just say that number one more time, $80,000 per day. So this 1 hour, I mean, divided by however many people we are, but that's still an astronomical number. And this is why I want us to focus quite a lot in the class on what we've been calling frugal oceanography. This is a very inhospitable place. Without this massive infrastructure, the system, almost a team of engineers and safety officers and captain and cooks, we would die here pretty much instantaneously. So to be able to do science at sea requires this massive infrastructure, which is very expensive. That's why the analogy to space is also very interesting, because just like astronauts, it's an incredibly inhospitable place. Uh, and especially when the scale and the large platforms are needed. And so small scale autonomous platforms dramatically reduce the cost. And we'll talk a little bit about that. Daniel, were you going to say something? Sorry. No. Okay. Um, so that's an important piece of the puzzle. I think one thing that we will do is just sort of when I look at statistics and other sets of factors, it's fairly easy to think about this, but the ocean is the largest biosphere on the planet. I mean, everybody talks about this 70% surface area, but what they don't talk about is the volume associated with it. So you multiply the surface area by the average depth of 4 kilometers, and that's really when you start to see the scale. And often enough, we have thought that in the old days, it was believed there was no life in deeper waters. But this volume is a huge critical challenge in our measurement right now. And we'll talk about this in context of microplastics. Uh, Surface, which is visible by satellites, is a very, very small, it's almost like measuring the skin. If you were treating a human being and all you could do is make measurements on just the skin deep, 2 micron layer of a person's skin, and that's it. And so majority of the measurements, at least satellite-based measurements, are kind of skin effects. Which is really difficult to understand because of the volume associated with the ocean and then the depth associated. And the deeper waters you're in, harder the measurements because your tools have to survive incredibly high pressure, incredibly corrosive environments. So depth is a critical piece of this puzzle. And most of the time, the depth is really where we know very little about. And kind of one way of thinking about this is just the picture that I want you all to keep in your mind when you're thinking about the ocean. This is a painting that an artist in the lab made, Rebecca. Uh, and Rebecca and I were talking about canyons. So when I go to the ocean, I often like to feel and imagine that I am actually in the Grand Canyon. And so imagine if there was a chasm that you could cut in the ocean. That's really when you would see this sort of a living ocean play out. And that's what's really hard. So it's the volumetric measurements around this that become really critical in understanding many of the processes that undergo in the ocean. So I want you to remember this picture. And there's a very famous line that Rachel Carson, I'm assuming everybody's heard of that name before, Rachel Carson, right? Uh, actually, curious, show of hands, how many of you have heard of that name? So quick homework, if you have not, please, please, please try digging that up as a name. And bonus point if you pick up one of her books and read it. She's most famous for Silent Spring, which literally created the EPA in our country. Like, and again, today and now is the time to really think about her writings. She was an incredible activist writer, so she really fought incredible amount of environmental laws and everything. But very few people know that Rachel Carson was trained as a master's in marine biology, and before working on many of these environmental issues, she wrote incredible books about the ocean. And in one, she describes this idea of the reason people don't emotionally engage with the ocean is because it's a little bit, there is a veil that the ocean is wearing, which is this thin layer that when I look at the ocean, light bounces off and you just think of it as a non-living surface. And this chasm sort of changes that viewpoint that it's only if we could see inside the ocean, it would change our perspectives around that. Okay, so another challenge associated with this is I love this animation. So you're going to see this play out on the left side. This is a simulation of some of the biology that we know, some of the physical chemistry, some of the circulations that we know. It's called the MIT Darwin Project. And the PI on that was Mick Follows. And they took on this challenge to do high resolution simulations in the ocean, but to a certain extent of the biology. So I'm going to play this video for a second, and I want you to feel, oh, oh no, this is a really remarkable video. Okay, let me just dig this video for a second. Okay. So I'll just let you watch this video for a second on the left. Ignore what's happening on the right. As I said, this is a simulation. I think the voxel size here is maybe a kilometer by kilometer grid. It's smoothened. But what is being plotted is, in some sense, chlorophyll. And what's interesting about this is, if you notice Australia there that's passing by, you see seasons, the greening that's occurring. So this is seasonality on kind of what happens when trees grow and shrink as summers pass by. And similarly, at that same time scale, there is this incredible dynamics at play. And you suddenly realize that now our problem is not just, I described that the ocean is big, but now the second challenge is that it's dynamic. So just making one measurement is not sufficient. The moon is not so dynamic after all, or maybe it is, we don't understand it. But the ocean is incredibly dynamic. So I could be in one place and you said, so that's, you can see the little red across Sahara. This is basically because of the desert sands being pumped into the ocean per se. You can see red is high chlorophyll, so it implies massive amount of growth per se at play. But this is the second challenge now, that the ocean is incredibly dynamic. Now, this might sound like a cliche, but it's worth just saying this. Many of you probably heard it before, but I still want to say it. The ocean roughly produces, 50% of all the oxygen that's in our atmosphere. It's kind of debated in terms of how cyanobacteria, early on, there was only cyanobacteria in the ocean. So early on, 100% of oxygen, this is prior to plants. But now the bulk estimates are around 50%. But more importantly, it actually absorbs 90+% of heat. So if this heat absorption was removed, you would be in big, big trouble to begin with. And I think this is why instruments to be able to measure temperature as precisely as possible in the water bodies has been very critical to begin with. Something that we work on as a lab, which would be really fun to think about in terms of projects, is also that between 30 to 40% of all CO2 molecules that we are emitting as humanity are being absorbed by the ocean by two different processes. They're roughly called biological pump and alkalinity pump. But we'll talk about that process. But if somebody was to switch this gigaton scale carbon sequestration technology, our carbon dioxide in the atmosphere will go from 400 plus ppm to 600 ppm instantaneously. So you suddenly realize that it's a type of a service that it provides that's very critical in the current day and age on sequestration. And that is fundamentally at stake because we don't really understand how that sequestration process works. And I'll talk a little bit about that sequestration process itself in a little bit. So, I think not only it's big, it's dynamic, and at that same time, it has some critical global functions, even if you don't care about the biodiversity side of the story. It's also just the life as you know it on the planet would not exist without the existence of our oceans in their current state. Now we'll transition and talk a little bit about what has changed in the ocean. Uh, any question on kind of some of these larger scale implications before I'll talk a little bit about what is changing as a function of time via human activity?
I had a quick question about
Yes.
the animation that you showed.
Yes.
What creates the dynamic processes in the ocean? So the green changing is like chlorophyll with the seasons. What's the equivalent in the ocean?
Yeah, no, I think this is a very important point. Almost all parameters in the ocean are dynamic. So the first thing that you think about is some of you have done some of these bacterial growth curves and the log phase, and you see that things rise when you have lots of nutrient and then things die. So even in a small vial, you can recreate this dynamics of growth, stationary phase, and decline. Now, this is possible to see even in one species. Now imagine a cocktail of 1 million species all interacting with each other. I'm not even talking about any spatial structure. Imagine a well homogenized little vial, but with that much diversity. You'll imagine that many dynamics would essentially arise purely from that. Now we add it to the layer of ocean circulations to begin with, which are bringing new nutrients to one place, removing nutrients for the other place. There are oxygen minima zones that are growing. There is many effluxes from pollutants that we add to the ocean. So you start adding up this spatial structure and you start realizing that these simulations in some fact are really preliminary because they are writing very simple equations around what should a concentration of a biological species be as some parameter function. But when you talk about that much diversity, there is so much interconnectivity between these species. So predator-prey interactions, one of the projects that we have in the lab is thinking about this dynamics in a game theoretical perspective. So can we really understand some of this dynamics? And so some of you have read Nash equilibrium, for example. Nash equilibrium has been used to think about interspecies interactions and explore where that dynamics come from. Then there is another big open question of, is this dynamics, can it form stable points? Are there tipping points in this dynamics, for example? So if I was talking about the baselines don't change, but if I change the pH of the ocean exceedingly to a certain extent or temperature, which has already happened, how does that dynamics change? So in some sense, theoretically, we don't have the tools. I would argue, although the Darwin project is really remarkable, the biological complexity it incorporates is actually abysmally kind of amateur for good reason, because otherwise we don't even know how to simulate these things in a small while. So try doing the best known simulations on interspecies interactions have come from a group at Princeton almost 20 years ago, and the number of species they could treat was 3. So that's the best scale scenario where we are. We don't even have theoretical tools, but this is the best we can do currently without the class of measurements. And one way of thinking about these sets of dynamics is some of them are natural and some of them are also human induced. So when you see these giant red pieces right next to cities, this is actually waste water from the cities pumping all kinds of effluxes. And this runoff is essentially called the eutrophication as a process. So I'll show you this, for example. This is the worldwide map of, and I don't know if some of you remember what happened in 2019 and a couple years ago in the Bay. It was called the Bay Apocalypse. It was the largest number of dead mammals and fish and animals found in the San Francisco Bay because of this massive toxic bloom that had arose. And many of the sites that you're seeing here are sites in which ton of runoffs, human waste runoffs, many of chemical runoffs from cities kind of create an imbalance in nutrient availability. So this set of dynamics we are also perturbing, and some of that dynamics is actually intrinsic, and some dynamics is perturbed by human activity. There's all kinds of questions really around. Another perturbation that's important is it's an interesting thing to think about in terms of spills, and oil spills is a classic thing. And I find it always quite surprising. It's of the order of the amount of oil spills that we're really talking about something of the order of 10,000 spills in the ocean, roughly an average of 50 to 100 years lifetime of some of these spills and their impact. So then these are other kind of point perturbations that we make in the ocean that happen all the time. I mean, these accidents are far more common than we actually think about. Another big part of this perturbation that we know very little about is the plastics. I know many of you have mentioned and talked about plastics, and I would love for us to choose a few parts of the ocean. So even 11 kilometers down, people are seeing plastic. We're seeing plastic in birds, we're seeing plastic. And I think my direct observation around this is for the last 10 years, I've been out to sea. Every time we pull out a sample from the ocean, no matter what, it's filled with microplastics. So it's to an extent that it's always there. But there is a kind of a paradox and a puzzle which is important in here and which I want to really describe. So first of all, some of you have heard about the garbage patches. You know, these kind of figures make it appear as if you will arrive at this place, you will find a garbage patch. We're not that far from the garbage patch even right now, but it's not as if I'm going to see floating plastic. The density is still low. It's a lot of plastic, but because the density is low, it's not trivial and it's not energy efficient to remove it currently. So there are lots of efforts of just going out with like these massive surface skimmers. The problem with that is it's not as if these cartoons make them appear that, oh, we just have to go to this one place and clean it. But that's not, if you were at this place, you wouldn't see a massive number of things. So just a ton of effort and energy is spent on removing a piece of any foreign object from the ocean. But there is a puzzle to this. And the puzzle remains, which is 99% of all plastic is still missing. And what we mean by that is that there are very good estimates of the kind of amount of plastic that we dump. It goes into the ocean. But then when we characterize the places and total volume that we find, it is actually a very small percentage of what we have dumped from the estimates of rivers and things that are dumping plastic. And there's been a lot of work that's done on this, and a very simple, but the complexity of where this comes from in a theory that's out there is actually this idea that traditionally plastics have a density that is lighter than seawater. But then when it forms into microplastic, there will be life that will grow on that plastic, and that will change its density. And then that microplastic will sink to the bottom. But when that microplastic sinks to the bottom, it is not physiologically viable for those organisms to remain at that depth. They die off or they leave, and then that microplastic rises again. So going back to Ragad's question about dynamics, even microplastic has this dynamic circulation that's at play. So whenever we do surface sampling, what has been a problem with microplastics alone is it is also naturally become a volumetric problem. Removing something from a volume is very, very hard compared to removing something from the surface. Even though much of the plastic that we throw in the ocean, if you would notice, if it doesn't have air, even without air or anything, it floats. But when it breaks into small pieces, when biology takes over, it becomes heavier and then it goes in deeper cycles. Was there a question? No?
Oh, I had a quick question
Yeah, go
actually.
ahead.
Um, so you're saying that the organisms grow on the microplastics and then it sinks and then they die off and it rises again. Is
Mhm.
this a natural process that exists without the plastic? Do they tend to grow on something else and then this process still exists, but it's just happening on plastic? Or is it being caused by the particles being there?
Yeah, I think none of this is published. There's a group that we've been working with in Naples. They have really focused on this problem per se, and so this is sort of the first sets of evidence. But this is a natural process. It would also happen for many other things. But with microplastics, the density is uh, the difference between seawater density and microplastic density is small enough that microbial growth and biofilms on top of it can topple. But if it was a piece of styrofoam that's very, very light, no amount of growth on it will actually sink it. But yeah, I don't know how to think about whether it's a natural process. I think uh, any substrates that's on in the ocean, things grow on it. You put a boat in the water for a month or a year, stuff grows on it. Everything grows on it. When we put clay, bacteria will grow on it. So there is always what I mean, the Navy term, it's called biofouling. There's always biofouling. But it's also an interesting, I mean, a lot of people have been thinking a little bit about sequencing and sampling which organisms are using plastic as an energy source. There are certain sets of ideas around, but again, the problem is that reaching the plastic in the ocean is actually quite hard to begin with because it's become volumetric as a problem now, not just a surface effect. And the biggest initiatives around, even if we were to massively scale them up, are only tackling the surface side of and again, even then the energy, if some of you want to do the by the numbers, it would actually be very interesting for you to do the energy calculation of how much energy would it take to remove all of the surface plastics in the ocean. See whether those dilution factors and numbers actually work out, because we have very good numbers on energy on shipping and ships. So you could combine the two problems to see whether that solution makes sense. Um, Okay, any other comments on the microplastics? And we'll come back to these sets of ideas over and over again. But I just want to give you kind of a broader survey of why these sets of challenges are hard. A significant portion of plastic in the ocean is actually fishing gear as well. So it's sort of they are so you can think about regulation as a very interesting approach, really explicitly thinking about how do you make fishing gear far more environmentally friendly. This is one of the biggest issue because small scale fisheries have very little resources, and that's 50% of all fishing. So this is kind of another very important space to think about. Let's just take a look at temperature for a second. Temperature is a important, interesting analogy in terms of I had said 90% of heat is being absorbed. This is slightly older data, but if you just look around currently in all of the data sets, the fastest temperature change on our planet has actually occurred in Arctic and some of these polar areas. So just in terms of delta T. And many of these sets of oceans are now warming up to such an extent that organisms that we used to find in tropical zones are actually moving up significantly in latitude because now it's viable for them to live in higher and higher latitudes. So temperature anomalies and kind of heat waves is a very common thread. And why these measurements are important and why it's risky is also just the fact that temperature gradients drive some of the largest circulations in the ocean. And what I had said about tipping point is the idea that if these circulations are perturbed, you will get a completely different ocean. And that has a huge implication on humans in general. So some of you have heard about this, that there are these giant Gulf circulations that keep stable temperatures. So heat perturbation is a very important. And then again, it goes back to the notion of how we measure temperature. And with that, it's worth a moment to actually tell you a story about temperature. How many of you been to Hopkins Marine Station? Sorry, show of hands. Only two people. Kevin, you've been to Hopkins, no? What? You haven't been to Hopkins? Oh my goodness. Okay, another homework assignment. Figure out how to go to Monterey. Danny is going there every other week or something.
Every
Hopkins.
Thursdays and Fridays every week.
I'll put Danny on the spot here. This is an open invitation from Danny. Anybody who wants to go. It's very important because we as a university has a remarkable marine station. And I'm very happy actually. We could do a class trip, and especially for people who will choose ocean as a project, but in general. So Kevin, can you write that down somewhere? One thing that would be useful is in one of the Fridays, and we can actually go all as a group because it would be very important to understand on the ground. Um, Marine stations are critical for studying, and I think related to temperature, I wanted to tell you about something, uh, which is both a funny story, but has a tradition associated with it. And this temperature measurements. Hopkins Marine Station. There is a picture that I am. Okay. Just as a marine station, we are the oldest marine station on the West Coast to begin with. And the thing that I wanted to show you guys is because of that, there has been, let's see, there is a photo that I'm looking for. Temperature measurement, Hopkins has a surprise. I can't find the. Hoping I can find this photo. If I can't find the photo, I'll describe it. Okay, it's taking me too long to find the photo, which kind of kills the story, but it's worth okay. Longest temperature record, Hopkins Marine Station, daily sea surface temperature. Okay, I think I'll just describe it. Looks like that photo is not, I can't find it. So for the last 100 years, something very interesting has happened at the Hopkins Marine Station. There is a person who's called the Guardian, and this is kind of talk about frugal science. That person lives on the station every single day for the last 100 years. Somebody takes a bucket that is also from that same time, goes to a specific spot at the marine station, pulls out a bucket of water, and using a thermometer makes a temperature measurement. And it is an incredible time series of every single day temperature measurements. And it's actually really worth going and seeing that at the station. Uh, and this is done by graduate students that live on the station. So you become in charge of this longest time series. But from a technology point of view, it's very, very simple. It's just a bucket. So anybody, half of the world's population lives close to the ocean per se. And so people that live literally on the water could have done this. And in some sense, this is also the rigor in these measurements with very simple tools if we could do something like that. And it's funny to watch and read that record per se, where the person writes a few notes about how they felt exact same time. And there is date. You can go back all the way 100 years ago and see what that person was doing and saying. So viscerally, it's quite fun to watch that, wait a second, we don't actually need that much sometimes to create, but what we need is longitudinal context. That's what's so critically missing, is that for dynamics and change, you want simple measurements that have been made for a very long period of time. That's really kind of an analogy to this bucket. And at some point of time, I'll find that bucket and I'll show that with you with the actual notes. There is a register that's physically somebody writes that temperature. And if we go to Hopkins, we can go and see that register. It's kind of important because it will give you a sense of historic records at play. So that was a little bit about temperature. And then maybe another platform that I want to describe, which is rising quite a lot now from a technology point of view and on solutions point of view, something that's been quite a game changer, is two things. One really is satellites. So, satellites are important. There's a lot of things that satellites can currently measure. And when you think about this, it's quite surprising to think about, but currently satellites can do temperature estimates, they do current estimates, they do waves, they do ocean height. Of course, they do chlorophyll. They can also do spectra of sets of particles that are in there. So when Sahara Desert blows on, you can actually see that in the satellite. There is some amount of physiology that's also in play. And now there are hyperspectral satellites that are being launched that are trying to also do many different wavelengths. So this has been the primary growth in our data measurements in the ocean, has really been satellites. But the big challenge with these measurements has been that they are surface measurements. They don't go deep. There is a second platform that I just want to briefly mention, which is important in terms of thinking about sets of projects and other things. And this is called the Argo Float. And Argo Float is a really interesting solution to ocean measurements. I was just talking to one of the groups at Ambari yesterday about the Argo Float program. I think they have of the order of Actually, I have to check live how many Argo floats. If we were to do live Argo float dashboard, you can all see this. So if you go in here, you can watch. These are these tiny little, they're not so little, they are height of a human being, and it's loading right now. And what they have the capacity to, first of all, they're totally autonomous. Majority of the Argo floats are making physical measurements, and they sink to a certain depth, say 1,000 meters. They make the measurements during their sinking, then they have a timer, and then they rise back up again. They sink, they rise back. This is funny, the dashboard isn't online. Let's try that here again. So if this dashboard was live, you would actually see live data from these sets of instruments coming in. And this is one example of our coverage. And at this point, in the old days, the only way we should be able to make the measurements was through ships making transits. But now the data sets from Argo floats is actually 3 to 4 times higher in both coverage and numbers as compared to ship transits, at least in physical oceanography. Biological sites of measurements are still really majority done by vessels, but it's a very important platform that has grown as a community for a while. But so this is what people do. They will dump this thing in. You can see the size of this, walk away, and it has a transit time for every 12 hours. It'll send a ping and give you a depth estimate. So if you want for your by the numbers, you can go to the ArgoFloat website, download all the data, play around with it. But this is the cost per float. So I had said, say 10,000 floats currently exist. This is 118,000 per instrument. So let's call it 100,000. You multiply 100,000 by another 1,000, this roughly between 4 to 10,000 of them. So this is 10 to the power 5. You add another 4 to that, that's 10 to the power 9. That starts to give you the cost. They have a lifetime of 5 years, so they have to be continuously replaced because when the battery dies, it sinks to the bottom of the ocean. So kind of, I think one very important thing in this framework to think about is what does cost and instrumentation of something like this look like? There are some smaller instrument packages people have been building, but they don't really do what Argo Float can do. But this is a tremendous space to think about because it gives us a sense of asking, when you think about these types of networks and infrastructure, why cost is critical. We still need 100x more coverage of the ocean, but that wouldn't be feasible from a financial point of view, for example. And this is also why when you're thinking about autonomous measurement platforms, they have to be much, much more frugal. So kind of what happened in the drone industry or what happened in the 3D printer industry never happened for the ocean as yet. But there is a lot of talk now that people are excited. And some of this is also being compromising in certain sense of thinking about what is the actual granularity of data that we need, because this by itself is a little bit tricky. So there is one of the platforms that's called Goose, which is the global measurements platform. And what's valuable here is that it is now combining all kinds of data sets. So now you can see there is not just Argo. Last time I looked at this data, 4,000 Argos. There is a deep Argo, there's a bio Argo. Bio Argo has chlorophyll now in it. But now there are these other sets of platforms. There are certain sets of time series in it, which is done by ships. And there is a single platform that combines all of this data set. So if anybody wants, you should just first take a look at Goose website. It'll give a sense of thinking about what type of data many of these things are collecting. And then I'm going to close by just mentioning another, actually, yeah, maybe two more examples, and then we'll turn this into a discussion because I want to spend some time talking about open problems in this space. Another interesting thing about the ocean that's important in measurement is that it's dynamic in space and time, but in many orders of magnitude. So there are certain sets of things you want to measure instantaneously. So x-axis here is spatial scales, y-axis here are time scales, and there are certain sets of things you want to measure in smaller and smaller resolution. There are certain other sets of things you want to measure at high resolution, both in space and time. And that's also an incredible challenge. So Ragad, when I had said that the ocean is dynamic, it's dynamic at all of these length scales. For some question that we care about, we have to really ask that the frequency of measurements have to be very fast. For others, they also have to be at the scale of 100 years, 500 years, and that's hard because when you're designing something, when you're doing some very fast frequency measurements, and you also have to cover certain sets of time scales, this becomes very problematic. Another historic, I'll kind of end the, I want to turn this into a discussion now, but time for another story, which is you might feel sort of helpless in this, and you might say, what could actually be done about it? This sounds unsurmountable. And this is where these examples become important. There was a remarkable oceanographer, and his name was Alistair Hardy. He was in Plymouth, UK, and he had invented all kinds of techniques. He was out in the ocean all the time. And this is now, I'm talking about 1940s, 1950s. And he had an idea that to reduce the cost of ocean measurements, we should actually take advantage of the shipping fleets. So shipping fleets, 70% of everything that you own probably came through a ship. So think about that. I mean, it's a massive amount of shipping traffic on our oceans. And he invented, which now happens to be the longest time series of ocean records. It's called Plankto. Does anybody remember the name? I'm blanking on the name. Let's actually, plankton recorder. Yeah. Like a tape recorder, he named it a plankton recorder. And I just want to show this. We're going to watch this video for a second. So what you're looking at is a pretty clever device. There is no battery in this thing. It's a giant chunk of metal. It attaches to the back of any ship via line. And inside there is a silk reel. Imagine a very long reel of silk, or like a sari, that's rolled between two rollers. And as the boat moves, it uses the thrust of that water from here. You can see this little nozzle. That water flows through. It uses the thrust of that water to rotate these wheels. And the plankton and the microbial life in it gets squeezed between these two reels and stored like a roll. So when you arrive, this can be out there for 3 months, 4 months. And then when somebody arrives, they essentially get this massive roller. And so in UK, there is a very large stack of these things that are actually operating. Okay, I think the Argo floats just came live. Anyway, you guys should look at it. It's super easy to look at the site. But I want to show you what the processing of the plankton recorder looks like. Continuous plankton recorders. Let's just look at images. So this is what I was mentioning. Totally passive as a device, rolls in. There is a fixative solution that fixes it. And just like the archival scrolls, you basically have these massive scrolls inside. And now you can imagine the hard part is it's like insane to process it. So we've been thinking about a project of a digital version of something like this. So if somebody is excited in the project, We can build one of these plankton recorders, but you can imagine that we have to digitize all of this and we have to think about imaging associated with these sets of scrolls. So currently these scrolls are processed by all kinds of people, and then this is what that looks like. So there is a room somewhere in UK which literally has stacks and stacks and stacks of these rollers sitting somewhere, and with people going in, and you can go back. I often think about this as a fossil record. You could go back and pull out this from a ship cruise that went from Europe to the US and look at and say, oh, this was the corresponding point in the roll is the location of the boat. And from that, you can infer what was happening in actually the water column at that time. So it's one of the oldest plankton continuous record that we have so far. It hasn't been digitized, so its impact is not in the same scale as Argo. The data isn't really available. So one project I've been thinking a little bit about is after 50 to 70 years of Hardy's work, reinventing this tool in a digital era. So folks that are kind of interested will sit down and talk about this in a lot more detail. But I think I wanted to mention that because you sort of suddenly realize that this as an innovation has actually scaled, and 100 plus shipping vessels around the world carry these things continuously. So right now there is a boat somewhere that's actually towing it. And in some way, uh, we will talk about Planktoscope, another project that we released in the lab, which now has around 500 units floating around the world, completely open source. It's a digital way of recording what's in the water column. And it was the same idea. It was inspired by this notion that not just scientists, but citizens. So we targeted sailors first because sailors go out, and there's 15,000 or so sailing boats out at sea. So one of the important lessons from that is you have to think a little bit about what other infrastructure that already exists that we can leverage to increase our capacity. Frankly, there is an incredible amount of military infrastructure that's there. And I kind of argue when I think about it is they're using our oceans anyway. They should be required to have all kinds of, and there is no cost limitation that they have. So there must be almost, so we've talked to the French Navy. French Navy has some planktoscopes now as a trial program, but you can imagine that in the choice of decision of designing of much of this military hardware, there is also an added thread about scientific measurements being made with the data being publicly available, because majority of the time, many of these vessels at sea are out there, but none of the data comes through. So thinking about this, in some sense, it's valuable to also just explore what infrastructure can you take advantage of. Okay, for the next 10, 15 minutes, I want to turn this into a discussion per se. Any sets of ideas that pop in people's mind, any sets of problems, I just described a few sets of things. What comes to mind? Any sets of questions around this as a framework? Kind of open mic. If you could measure anything, what would you measure in the ocean? No bounds. No. Or what infrastructure exists that we could leverage that we're not leveraging currently?
Maybe like an oil rig.
Oil rig. I'm so happy you said that. I have this proposal that I wrote. It's been rejected many times to private foundations to buy a decommissioned oil rig. It would be phenomenal to have permanent research stations. The reason for this is very simple because oil rigs, the infrastructure is already there. It's not safe to operate. They've already drilled the oil out. And actually, we even estimated in terms of cost associated with it. The framework is that by some amount of presence and some instruments being there, some of the safety of that can also be checked, because otherwise they have to go back and keep checking again to make sure that some of the pipes are still closed. It hasn't led to an environmental disaster, for example. The other point around the oil rigs is that we have many of these floating infrastructure. They are in the Lagrangian frame of reference because they float with the water. So over time, they all start accumulating in some places. So on the other hand, stationary measurements are very hard to do in the ocean because you need a vessel. So that's very valuable. And then it would just be super cool to have. And, you know, it's a little bit ironic, the oil that caused the problem, if they were required to, that everybody who has dug oil from the ocean is required to have X number of units of oil rigs. So yeah, I think if people are interested and some of you have a design bent, I'm happy to send you guys the proposal. One of the things that we've been excited about in the oil rig context is also using them to be able to also do deep measurements, because already there is some amount of infrastructure on oil rigs when they were built for the piping and other sets of scenarios. Oil rigs are also very common in other places where there are other functioning oil rigs, which becomes very important for sensitive measurements around leaks and other factors. Because when leaks do happen, there is a much long time. There are some measurements that have been made, but we know very little about how oil disappears in the ocean. That 50 to 100-year time scale is a microbial time scale. There's some really beautiful papers that bacteria chew up, and there is an interfacial instability that drives that. There was a science paper a couple years ago. But I love that comment, and it's wind would be another one of those examples, because some amount of wind infrastructure on coastal areas is also being put. In fact, Argo floats are only put in deeper waters, not in shallower waters. So shallow waters are actually less sampled, and there is lots of shallow water rigs out there too. So it's also complementary in some sense.
On
Go ahead,
the
Dan.
on the plastic note, I would be intrigued in thinking about some, I don't know how it would work, but some like magnet, obviously plastic is not magnetic, but something that would draw the plastic to it either through charge that you could
Yes.
draw through and pull the microplastics, and a way of
Yes.
attracting them
Yeah.
would be
No,
interesting.
I think it's a super cool idea. One thread to think a little bit about this is understanding the surface chemistry of what's happening on the microplastic itself would be the place to see where that concentration mechanism can come into play. For the longest time, I've been working on one idea for a while on kind of surface interactions. The problem really boils down to the total volume we're talking about is really immense. So in some way, the challenge lies in choosing very high density areas firstly for microplastic, and using that as a way to compare. And I think the hard part about, I mean, the biggest critique, there have been some very strong scientific critique on the surface skimming to remove microplastics in the ocean, because there is a significant portion of ecosystem that lives on the surface layer. It's called the Knudsen layer. And most of those species literally die. Surface of the ocean is not a dead. It's live, it's very unique. And this is the part of the challenge of thinking a lot more about, when you think about volumetric threads, filtration is not an option. So it has to be very specific to microplastics per se. So Danny, I have a couple of ideas in that space. And one thing that would be fun to do for you to choose the by the numbers assignment purely on microplastics, so that we just first start with the back of the envelope calculations of what does the energetics of volumetric removal of microplastics look like. And by the numbers is sort of an approach to just keep us honest. it's best for us to know that, oh, by the way, if we were to use this scheme, the total energy spent would be the entire energy budget of the world. That's good to know, because then he feels like, yes, I mean, that just is the starting point is there. We have to now think of something else to be able to appear, or it would take the lifetime of the universe to do it by just the transact ships. And so I think that's kind of the idea of by the numbers is an approach to sort of add teeth to some of these broad ideas. Other ideas that pop in people's minds, hearing about the ocean side of the story.
Just to chime in, is there
Perfectly, this
anything
is totally
about
open discussion, so you don't have to worry about it being a well-formed thoughts. Open mic is really just sharing what's on your mind. Go ahead, Kevin.
is there anything in the earthquake prediction space about, like, I know for example Japan is next to a fault, California is next to a fault, but maybe ocean floor like plate contact, maybe there's something on the longer time scale that can be measured. I don't know.
Yeah, I think two things come to mind with what you're mentioning. Firstly, totally unrelated because you mentioned Japan. Everybody remembers Fukushima, the disaster. Those disasters are also an opportunity for measurements because that radioactivity is now found, and people found debris with that radioactivity all the way on the California coast. And from that, they were able to figure out the transit time for these circulation currents. So that's just kind of funny things around. The most famous one, does anybody know the Duck Project? Some of you know that. There was a very famous disaster that happened that a shipping container lost billions and billions of floating bathroom ducks. You know that little yellow duck that we all love? It was coming on a shipping container, and in one spot they dumped, and it's a perfect tracer experiment because they float in the ocean. And so over the next 30, 40 years, people have been tracking, and it was the first time we could build measured circulation models for the ocean because that accident happened. So now, kind of what I'm evoking with this is clearly you should never do that. That is bad for the ocean. But now, when it has happened, how do you use that approach to build a frugal? Because the cost of that has happened, and building an architecture around measurement. So, what you said just reminded me of these two examples. Brilliant scientific examples. And now there are simulations that have been built and improved only because of the data that came from the Duck project. I think on the fault lines, there is an interesting analogy. I don't know if anybody's done this, but there was a paper 2 years ago where people looked at anomalies in internet traffic and kind of fiber optics that are underground. Like I am talking to you, actually, this might be satellite. It is satellite, but at some point of time, it goes underground. And majority of our internet traffic is coming through cables and repeaters that are under the ocean. And they were able to correlate and show temperature and other sets of variability that can be measured by that network. So if somebody is interested in for the by the numbers, take a look at that. I think those are real-time networks. So something like shocks on the bottom of the ocean floor would definitely have stretch in the fiber or other things that could be measured because they're very high frequency. And the kind of an event you describe is also very high frequency event. Frequency in the sense that it occurs in a short period of time, for example. So that's fiber optics definitely comes to mind. It is definitely, if any of you have interest, I know one of you is from the aerospace world. One thread to think a little bit about in that space is underwater communication is very, very hard. So comparing to what we do in space, and optical fibers is definitely the only system I know that is permanently kept and is providing continuous data. And there's a lot of potential in thinking about what could be done with an already existing infrastructure around it. Other ideas, comments?
I was thinking about like the temperature measurements and how they could be maybe correlated with disease, like infectious disease progression in different areas, because I know there's been a lot of work on like predicting where dengue is going to pop up, for instance, as like temperatures rise and like there's more
Yeah.
rainfall and all of that. So I wonder if there's been a connection between the ocean
Or
and
the ocean. That's an interesting, I do not know. I love that idea because I know somebody already mentioned it, either it was Danny, of there's a lot of new work right now on new parasites, and especially parasites for some of the critical organisms that are being monitored. I have a friend in Santa Barbara. He's almost retirement age. He's studied parasites in the ocean on the longest time. And he told me a trick. He said, Manu, if you want a new species that you want to name, the best thing to do is to find fish in the ocean and just dissect. So all of you who eat sushi, we're all eating fish parasites. It's the best way to identify uncharacterized invertebrates is fish parasites because there is such remarkable evolution that has happened. So kind of that's where my mind is rolling. There are microsporidia-like infections that occur in shrimp farms. Many of them are temperature sensitive. That's like $1 billion industry that gets impacted. So fungal diseases would be another one to think about. By the way, there are fungus in the ocean. Nobody talks about it, but there is a remarkable biodiversity of uncharacterized fungus. I know one of you works in a food fungus lab. Is that Peter? And Amy's lab and I think UNC has characterized a lot of new fungal species. But I'm wondering in terms of the disease side, what does that actually look like for marine mammals, for example? And I don't think I've seen anything that correlates well with kind of trying to understand as temperature change. The most things I've seen in health, which Danny had talked about a little bit, has been really around the UV exposure, but also pH. Acidification, a lot of people are currently running physiological health measurements on many marine species as a function of pH change. So that's the largest change. And I didn't talk about it, but pH measurements are really critical in the ocean. Oh, go ahead, Danny.
Rod, we can talk definitely after class when we're next in person. My advisor and I were writing a review / perspective on how marine diseases are getting worse through climate change, through ocean acidification, warming, and pollution.
Awesome.
And then we're brainstorming a bunch of possible solutions for that. So that'd be really fun to talk about.
Yeah. And then I think Julio in Hopkins also does a lot of disease modeling in that space itself. Other comments, last minute comments, because I'm realizing we're almost out of time. I think it's important.
We have until
Oh,
12:20, so
I see.
yeah.
Is that true?
Yes.
I see.
Pretty
Yeah.
sure.
Oh, go ahead. I think one thing I just want to mention is it's important for you. I want to break the ice. You should feel this is safe space. And just when we have these discussions, the technical parts you can all pick up, you can read reviews and stuff. Much of the purpose of taking this class is really having conversations with like-minded people who feel empathy for the same sets of problems and use this to generate a massive number of ideas. So what I want you to do is whenever we're discussing these sets of things, to speak your mind, like just these are real-time brainstorming, and many of these brainstormings will then turn into projects. And it might be that you are the one that seeded that idea, but you don't have to choose to work on that project, somebody else, because it strikes in conversations are hidden ideas that spark a completely new thread in somebody else's mind. So don't feel at all like, oh, if I'm gonna talk about something, that means that's what I am tied to, or no. This is just free fall of ideas. Other ocean kind of things that pop in your mind when you think about. What about oceans of other planets? So I didn't talk about this, but there is a remarkable NASA program right now on trying to make measurements. Many moons of Saturn are fully oceanic, but they have ice cover, and that's pretty hard. And there's a really cool measurement I heard about from a person I was giving a talk in. They had put a mass spec on a space probe satellite that was transecting. And it just so happens that these moons on either Jupiter or Saturn have a ocean covered with ice, but sometimes there are explosions and it continuously ejects a spray of water out into outer space. And what this flying mass spec was able to do is to pick up these water droplets in its flyby, analyze, and say what chemicals were present in that ocean. So imagine this, when you have ice moons, they have a massive ice cover and thickness, sometimes even a kilometer. So even if we landed a probe there, it would almost be impossible to drill that much to get access to the water. But it just so happens that they have these, yeah, sneezed. It really is like a sneeze. And they have these massive holes that are continuously shooting out these crystal sprays. And it was, another example of a funny, and when I heard that talk, I was thinking about role of drones and thinking a lot more about certain sets of surface measurements on top of the ocean surface that could be made. And we have very little flying instruments on the ocean uh, anyway, but it was this notion of thinking a lot more about, oh, what does a technique like that look like for Earth? Because many, at least for folks that are interested in the microbial diversity, they get aerosolized pretty easily in the ocean. So if I just take a little piece of air above the ocean on a syringe, you can really catch a ton of bacteria just because they're floating in the air in the sneeze. Any other kind of threads that come to mind? Oh, yes, field trip. Let's do a planning for that. So take a look at this, see if you can fill out a date. Anybody who wants to go with Danny one-on-one or something, that by itself is useful. But as a lab, as a class, I would love for us to all just go together. And then Kevin, we can look for a date basically post 21st April.
Yeah,
Post 21st April will work.
sounds good.
Yeah. Okay, so I think maybe there's a lot more that I could mention. One thing I wanted to do was actually I'll pick this up next time, but just as a prelude, you all can read a little bit about because I will do a lab session on this, but there is a tool that we developed now several years ago, which I will kind of dive into the hardware side of the story. I mean, you all saw that as an object. There's a tool that we developed that is, we call it Planktoscope. This was the very first incarnation of this. This is a student in the lab, Hong Chuan, and I built this first version. Then Thibaut joined the team and we built the second version. This is what the tool actually looks like now. So if I go in here, it's called planktoscope.org. And the instrument is very, very simple. It has a collection of water. It has a flow cell. It has a pump. There is a Raspberry Pi, an imaging system, and a GPS. And that allows us to essentially continuously image the continuous stream to be able to quantify and identify what's floating in the water column. So this is sort of the continuous data set around that looks like. And what I'll do is we will build one of these units to give people a sense of the entire platform is open source. So literally hundreds of people around the world have now replicated this as a platform. And all the instructions of how to build, everything is online, but in a lab session, we will actually just assemble one of the units and I'll teach you guys how to build it. This was sort of our answer to thinking about the continuous plankton recording as a system. But what's been very interesting is that the platform has now grown massively. There's roughly around 300 units out there at this point. And it's a good example of thinking about how open source hardware can actually change. And this will also give people, if you're interested in more on the microbial side of the measurements, it's a good scenario to just learn what actually is out in a water column itself. So if you have a moment and you're interested in any ocean related projects, we will do the planktoscope as a case study. So dig in, read by yourself about the website, and then we'll do a Friday session where we literally build one from scratch. And anybody can in the class can also get trained on using them. We have many units. We have around 13, 14 units in the lab. And they're always evolving as a function of time. And at a later time, I'll talk a little bit about kind of what went into the design. If people are interested in the programming side of this itself, we'll do a lot more on the real-time analysis and ML layer to that as well. But this is sort of, it's valuable to think about some of these case studies of how we were able to go. So the existing platform costs roughly around $100,000. We brought the bill of materials costs from 100,000 to roughly around $600. And the kits that are now out there can cost somewhere between $1,000 to $2,000. So it's still 2 orders of magnitude of a decrease in the cost per se. And that's kind of has been what's been fun in the community to see how people have actually been using it. Okay, so that's all I wanted to say today. I'm gonna pick this up next week. We will do the by the numbers. We will also have the new cohort of international students join starting Monday. And one thing that I do want to do is for everybody, just have conversations amongst everybody to see if you're starting to form some cohesive team. I think we will build teams within 2 weeks from now because I like the idea of teams being there, even though the ideas are being refined. Oh, go ahead, Kevin.
Yeah, just on that thread about building teams, like please fill out the intro survey because what I've been doing with that is like making this like network graph of how similar your interests are. So that should help facilitate like your interests. For example, like Danny Collins and Peter Allen have the weight of this edge shows that maybe you should talk to each other if you haven't already. So just a reminder in order to help facilitate team making to fill out the survey.
Yeah, and I think it's the other one is also on Notion. We can start writing what we call the idea board. Idea board is like Danny has clearly expressed very deep interest in microplastics. So then create a little idea board around microplastic, then other people can start we can start recruiting each other to what you're passionate. It's an important part of team building that you have to be vocal champion of the problem that you care about and so passionate about it that somebody else would be like, oh, this is such a cool problem. And so like this is a part of almost recruiting each other in ways and forms. And I think ideally the main team that you will join will just be one team. So no fractured teams. You can be helping out and engaged in multiple teams, but it's useful to make a commitment to one team. But start talking about that amongst each other because if we haven't formed teams by 2 weeks, I will kind of force the team formation just because this is a project-driven class and working together is what I care the most about. Okay, any last minute comments? Otherwise, we're gonna call it a day.
How big are the teams?
I think it can be as small as 2 people, largest team, maybe 4 people, not more than that. I think, I mean, I'm not against, I just feel when there are 2 large teams, it has to be where everybody sort of really feels ownership. And I can imagine sometimes there is the small team dynamics is actually quite fun. I mean, many of you work on projects all the way where you work alone to working with 30, 40 people sometimes. It's just, I like the idea of 3 to 4 is an ideal number in what we have seen in the past. And again, it'll also not just be this cohort, there will also be the broader cohort to choose from and engage, and you'll be recruited to other teams and the other way around. But having conversations amongst each other is what's important. So whenever we cover a problem space, find time to have conversations with each other around that problem space. Because otherwise, I'll introduce a lot of different problem spaces and then they will just pass by and you can feel overwhelmed. But the best way to digest these problem spaces is to have conversations amongst each other. Okay, on that note, I have to run. I will see you guys again on Zoom on Monday and try having conversations about the ocean. Now you have 2 reasons not to sleep. So bye, everyone.